If you spend months growing crops but choose the wrong statistical layout for your field, your final data will be mathematically useless. We're going to bridge that gap today. We'll translate abstract statistical theory into a concrete, executable planting plan for your specific trial. Getting this right doesn't require a math degree. It just requires answering 3 straightforward questions about your seeds and your land. By answering these questions, you lock in the exact framework needed to ensure your analysis of variance, or ANOVA, yields accurate, publishable results. Step 1. Before we even look at specific grids, you have to verify that your experiment follows 3 non-negotiable rules. First is replication. You must repeat your treatments to get a valid estimate of error. Second is randomization. You must allocate treatments randomly to avoid bias. The third is local control, grouping experimental units into smaller blocks, accounting for natural soil heterogeneity. If your eventual layout violates any of these 3 pillars, the mathematical precision of your trial drops to 0. That brings us to our first decision point. Is your testing environment completely uniform, like a temperature-controlled greenhouse or an indoor lab? If your environment and your testing materials are perfectly uniform, this document dictates your choice. The Completely Randomized Design, or CRD. This design is incredibly efficient in these conditions because it is simple to set up and maximizes the degrees of freedom available for error estimation. Looking at this 4x3 layout grid, you can see how it works. Any treatment, like V1 or V4, has an equal chance of landing in any plot. But CRD has a strict vulnerability. Notice the pink assumption. The experimental units must be homogeneous. CRD works flawlessly indoors, but if you try using it in a standard, unpredictable farm field, natural environmental variations will severely compromise your data. So, for outdoor trials, we ask our second question: how does the soil fertility or moisture change across your specific piece of land? Farm fields almost always possess a gradient. They might get progressively wetter toward a low point, or more fertile moving down a slope. If your field has a gradient moving in just one direction, you require the layout shown at the bottom of this page, a randomized block design, or RBD. The core logic of RBD relies on orientation. You must place your blocks perpendicular to the field gradient. This ensures that all the plots inside a single block share the exact same soil conditions. But what if your field slopes downward while also getting sandier from east to west? When variation hits you from 2 different directions simultaneously, you have to upgrade to the Latin square design. This grid details how LSD handles it. You organize treatments so each variety appears exactly once per row and column, but there's a strict mathematical ceiling. As the notes show, LSD loses efficiency if you test more than 12 treatments. The physical geometry of your land dictates your choice. You will either control variation using parallel blocks or a rigid grid of rows and columns. That leaves our third and final question. Do you actually have enough seeds to perform standard replications? In early generation screening, breeders regularly have hundreds of different germplasm lines, but perhaps only 5 to 10 seeds of each. When standard replication is impossible, this page points to the Augmented Design. Here is the mechanical workaround. You plant standard check varieties, marked with a C, multiple times to estimate error. Your rare test genotypes, marked with a V, are planted only once. Another physical layout for this scenario is the hill plot design, which conserves space and material by planting seeds in small clumps instead of rows. A severe lack of material does not excuse a lack of rigorous science. Breeders simply shift the burden of replication onto their standard check varieties. You've evaluated your environment's homogeneity, mapped out your field gradients, and counted your available seeds. Because you followed these 3 steps, you now possess the exact statistical framework required for your trial. This field layout connects directly to your data. As this final note emphasizes, the ANOVA is the cornerstone of plant breeding experimentation, and it only works if the physical field geometry is correct. Draw the exact grid on paper, randomize your seed package accordingly, and begin planting. You can head into the field confident that your future data is secure.